Hello everyone, welcome to my channel. Today in this video, I'm going to start with the osmosis. So in the last class, I've discussed about water potential. Now, let's revise water potential before starting osmosis. What is water potential? Water potential is energy. We refer water potential as energy. What kind of energy? See, water is present in this. See, water is there in this. So if this is a pure water, so the water potential will be 0. Water potential क्या होगा? 0 होगा when pure water is present. Water potential क्या है? Water potential is a energy when the water molecules, there is a random movement of molecules. Random movement of molecules will take place, then there will be an energy, formation of energy will take place. That energy is kinetic energy. When in this solute will be added, when in the pure water solutes will be added, what will happen? Solute. So the water potential will decrease. Water potential will decrease. Why? Because the solute potential is always negative. So the water potential will decrease. So all the solution have a water potential, lower water potential than the pure water. So we can say that all the solution have lower water potential than the pure water. All the solution have lower water potential than pure water. Next, if when the pressure is greater than the atmospheric is applied to the pure water or a solution, what will happen? The water potential will increase when the pressure potential will be applied. If a pressure greater than atmospheric pressure applied to pure water or solution, its water potential will increase. So this we have already discussed in the previous class. So today I am going to start with the osmosis. See, osmosis, diffusion we have studied. So what we have seen in the diffusion, that there will be a transfer of molecules from higher concentration to the lower concentration, like that. So like diffusion, osmosis is a type of diffusion. What happens in this? The movement of water molecules from a solution with higher concentration, where the molecules will move from higher concentration of water molecules to a solution with a lower concentration of water molecules through a semi-permeable membrane. So we'll see what is semi-permeable membrane. See, if we know that a plant cell has a cell wall, the outermost covering of a plant cell is cell wall, and the second one is plasma membrane. We know that the cell wall is freely permeable. Cell wall is freely permeable. What does it mean freely permeable का मतलब क्या होता है? Means all the substances will easily enter into the membrane. सेल वॉल से हर एक वाटर सॉल्यूट सारे एंटर कर सकते हैं. दैट इज कॉल्ड फ्रीली permeable. But the plasma membrane is semi permeable. Plasma membrane is semi permeable. Semi permeable means what? Selective things will enter into the membrane, like water will enter, some solutes, sodium, potassium will enter into the membrane. So selective things will enter through the membrane, that is called semi permeable. So there are 4 different types of membranes are present, that is freely permeable, impermeable. What is impermeable? Through which nothing will pass through it. Then selective permeable. What is selective? Some selective molecules and solute and solvent will pass. So we'll see what happens in the osmosis. See, if this is a semi-permeable membrane, if this is semi-permeable membrane, what happens if this side, what happens in osmosis, this is a solute, here the water concentration is high, this side water concentration is high, and this side it is low. So the movement of water molecules will be from this side, from higher concentration to, it will move to the lower concentration, and this is a passively it will takes place. So osmosis is a process in which the movement of water molecules from higher concentration to the lower concentration of water molecules. So it will takes place where this side the concentration of water is higher than this side. This side is a less solute and this side is a more solute will be present. In plant cell usually a large central vacuole will be present. Plants में क्या होगा? This is a plant cell and this will be a large vacuole will be present, and this vacuole which contains vacuolar sap means some solutes and water will be present in it, contribute to the solute potential of the cell. So it will help in the solute potential, help in solute potential. In this what happen, this plant cell and the cell membrane cell membrane and the membrane of the vacuoles together plays an important for the determination of the movement of molecules in and out of the cell. So what will happen, this vacuole and the cell membrane will play an important role, and it will help in the movement of molecules in and out. In and out, how it will take? If inside the cell more solute will be present, then what will happen? There will be a movement of water inside the cell. If outside more concentration, lower concentration will be there, so it will move outside. It will help in that movement of the molecules. So in the last class, I have explained about this pressure gradient and the concentration gradient plays an important role. So pressure gradient and concentration gradient plays an important role for the net direction and the rate of osmosis. This will help in that. It will plays an important role for osmosis. When the water will move from its higher region to the lower region of so what will happen, until the equilibrium, it reaches to the equilibrium, the two will be, जब तक इनमें इक्विलिब्रियम, इनका मूवमेंट तब तक इक्वल नहीं हो जाए, इट विल नॉट रीच टू द इक्विलिब्रियम, इट विल टेक्स प्लेस. ये तब तक होगा जब तक इक्विलिब्रियम तक रीच नहीं कर जाते. Reach to that equilibrium, what will happen, the water potential will be same in both. In lab you have just you have done that potato osmometer. What happens in that, if you are taken a potato tube in the water, so entering into this potato through the osmosis, here water is there and in this sugar solution will be there. So what will happen, the water will enter from this tube to the potato and it will increase the level of water. One question is given, study the figure in which the two chambers A and B containing solutions and separately by a semi-permeable membrane. If this is a two chambers in A and B containing solutions and separated by a semi-permeable membrane, this is a semi-permeable membrane. If in this A the solute is less than B, so this is the two chambers, this is A and this is B, containing solutions and are separated by a semi-permeable membrane. So the question is, solution of which chamber has lower water potential? So which chamber will have lower water potential? This chamber A is having less solute and this chamber is having more solute. So as we know, if more solute will be there, so it will be negative. जितना solute present होगा उतना water potential क्या होगा, कम होगा. A में solutes कम है तो इसका क्या है, इसमें higher water molecules present है और B में सॉल्यूट ज़्यादा हैं. सो वाटर पोटेंशियल किस में ज़्यादा होगी, ए में. सो वाटर पोटेंशियल विल बी मोर इन ए. सॉल्यूशन ऑफ विच चैम्बर हैज़ लोअर वाटर पोटेंशियल, लोअर वाटर पोटेंशियल किसका होगा, बी का होगा. बी विल हैव लोअर वाटर पोटेंशियल. सेकेंड वन इज सॉल्यूशन ऑफ विच चैम्बर हैज़ द लोअर सॉल्यूट पोटेंशियल. इफ़ अगर हम बात करें, इसमें -5 है सॉल्यूट पोटेंशियल और इसमें सॉल्यूट पोटेंशियल -20 है, तो हम किसका सॉल्यूट पोटेंशियल ज़्यादा है, इसका, क्योंकि -5 और -20 कौन ज़्यादा हुआ, -20 ज़्यादा हुआ या -5. ऑब्वियसली -5 क्या हुआ, ज़्यादा हुआ -20 से. Third one is, in which direction will osmosis occur? So osmosis will occur from A to B. Why from A to B? Because here the water potential is higher than the B. So the movement will be from higher region to the lower region. At equilibrium, which chamber will have lower water potential? When these two will reach to an equilibrium, so A will have less water potential. Okay, we'll start now with the another experiment where solution of sucrose in which taken in a funnel is separated from a pure water in a beaker by a selectively permeable membrane. So we'll start with an experiment. Okay, let's see the experiment. What happens in this? This is a water. In this is a pure water. This is pure water. This is a funnel in which sucrose solution is present. Sucrose solution is present in this funnel, and this is a pure water, and this is selectively permeable membrane. and this we get it from eggshell. So how we can get this, we can make this membrane with the egg cell. We can remove the, if we have taken the egg, we can make a hole and remove all the egg white and albumin, yolk and albumin, through a small hole at the end of the egg and place the cell into the dilute HCl. And after few hours the shell will become semi-permeable. It will become a semi-permeable. So we have used that egg cell as a selectively permeable membrane. So what happened, this is a pure water and this is a sucrose solution. Through this it becomes a selectively permeable membrane. So this water will move inside, because here the concentration is higher, here the concentration is higher, and here the concentration is low because of the solute, and the water will enter into this funnel, and the water will move inside the funnel, and what will be the result, there will be the rise in the level of the solution. So this solution का जो है level वो rise कर जाएगा because the water is entering into the funnel. Till here it is clear. If in the next we'll see what will happen in this. If the same solution is there, this is a pure water and sucrose solution. If with the help of piston and we are applying force, pressure we are applying to this, what will happen? When the external pressure is applied from the upper part of the funnel, so that no water will diffuse. जब हम लोग external pressure apply कर रहे हैं with the help of piston, so what will happen? The water will not be entering into the funnel. See what happens in this, जितना solute potential होगा उतना ही हमें pressure apply करना पड़ेगा, so that will stop diffusion. जितना solute potential है, if solute potential is 20, then we have to apply 20 minus 20 is the solute potential. If you, as I told you, the pressure potential is always positive and solute potential is negative. जब हम लोग pressure apply कर रहे हैं तो क्या होगा, जितना solute potential है उतना ही हमें pressure apply करना पड़ेगा, so that it will stop entering the diffusion of water. जब diffusion stop हो जाएगा, क्यों stop होगा, क्योंकि जितना उतना ही pressure हम apply कर रहे हैं, so that we can stop entering the water into the funnel. So this will be 0. तो water potential क्या हो जाएगा, 0. जितना water solute potential होगा हमें उतना ही pressure potential apply करना होगा. The greater will be the solute concentration and the greater will be the pressure required to prevent water from diffusing in. So the osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. As I told you, this is minus, solute potential will be always negative and pressure potential will be positive. Now we'll see what is plasmolysis. So see, the movement of water will depends on the surrounding solution. सराउंडिंग में कैसा सॉल्यूशन है उस पर डिपेंड करेगा कि वाटर का मूवमेंट कैसा होगा. इफ़ द एक्सटर्नल सॉल्यूशन, फर्स्ट ऑफ ऑल, if the external solution, if this is a cell, the external solution balances the osmotic pressure of the cell. बाहर का solution कैसा होगा उससे डिपेंड करेगा कि साइटोप्लाज्म का osmotic pressure कैसा होगा. अगर inside the cell more water potential is less, तो क्या होगा, बाहर the water will enter into the cell. क्या होगा, endosmosis will takes place. If the water potential is low outside, so we'll see what is isotonic, hypotonic and hypertonic. To understand plasmolysis, we need to understand these three things. See, this is the three different types of solution will be there. Different type of solution, 3 different type of solution. What is solution? Solution is solute plus solvent. Here what is solvent? Water will be solvent, and here solute will be, it can be salt, sugar can be solvent. So there we have seen, we will study about 3 different types of solution. First of all, we'll see what is hypotonic. What is hypotonic? If, first of all, I'll make a cell. This is a cell. If the external solution, this is external solution, is more dilute than the cytoplasm. We know that सेल है, सेल में साइटोप्लाज्म है, तो ये एक्सटर्नल सॉल्यूशन है. सॉल्यूशन में क्या होगा? वाटर एंड solvent and solute will be present. If less solute will be present in the external solution, less solute, then in the cytoplasm, this kind of solution is called hypotonic solution. अगर बाहर solution में less solute है, then the cytoplasm क्या होगा? इसे बोलेंगे hypotonic solution. Next we'll see hypertonic. What will happen? In this hypertonic, the external solution will be more concentrated, then that kind of solution is called hypertonic. External solution में क्या होगा? More solute will be present. बाहर यहाँ पे more solute will be present. More solute, more concentrated. Then this kind of solution is called hypertonic. So what will happen in this solution? In hypotonic, less solute is there. Here the concentration of, in hypotonic, the concentration of water is more, so the movement will be from inside the cell. In hypotonic solution, the cell will swell because of the water will enter into the cell. Cell swells in hypotonic solution, and the cell, in this what will happen, the concentration of solute is more in this outside, so the water will, the movement will be from inside the cell to the solution. तो इसमें क्या होगा? Cell will shrink. समझ में आ रहा है कि क्यों श्रिंक करेगा? क्योंकि बाहर कंसंट्रेशन कम है वाटर का, सो वाटर विल मूव फ्रॉम हायर कंसंट्रेशन टू द लोअर कंसंट्रेशन, सो दैट the cell will shrink. And the isotonic, the third one is isotonic. Isotonic में क्या होगा? The inside the cell and the outer external in solution will be same. So what will happen? The जितना amount enter करेगा उतना ही amount water का बाहर आएगा. So in this almost it will be equal than the cell. एक्सटर्नल सॉल्यूशन और सेल का जो है सॉल्यूशन, सेल के इनसाइड एंड आउटसाइड, इट विल बी इक्वल. So when the cell will be placed in the isotonic solution, there is no net flow of water towards the inside or outside. If the external solution balances osmotic pressure of the cytoplasm, it is said to be isotonic. क्या होगा? अगर एक्सटर्नल सॉल्यूशन बैलेंस हो जाए और तो इनके बीच का जो ऑस्मोटिक प्रेशर जो है साइटोप्लाज्म का, उसे बोलेंगे आइसोटोनिक. और जब सेल एक्सटर्नल सॉल्यूशन और when the water flows in the cell and out of the cell and they reach to the equilibrium, the cells are said to be flaccid. When they reached equilibrium, to be said flaccid. Now we'll see what is plasmolysis. Plasmolysis क्या है? This plasmolysis occurs when the water moves out of the cell and the cell membrane of a plant cell shrinks from its cell wall. Plant की जो cell है, plasmolysis occurs when water moves out of the cell. साइटोप्लाज्म में जो water present होते हैं, when it moves out from the cell and the cell shrink कर जाता है. सेल का शेप कैसा हो जाएगा? श्रिंक कर जाएगा. दिस इज अ सेल वॉल. सेल वॉल श्रिंक नहीं करेगा बिकॉज इट इज मेड अप ऑफ सेलुलोज. इट विल नॉट श्रिंक. व्हेन दिस हैपन्स, सेल इज प्लेस्ड इन अ सॉल्यूशन. हमने इस सेल को एक सॉल्यूशन में रखा है एंड दैट सेल इज, क्या होगा? हाइपोटोनिक सॉल्यूशन में रखेंगे या हाइपरटोनिक सॉल्यूशन में रखेंगे? वी कैन कीप इन द हाइपरटोनिक सॉल्यूशन. इसमें रखेंगे, हाइपरटोनिक सॉल्यूशन में रखेंगे तो क्या होगा? सेल से, क्यों हाइपरटोनिक? बिकॉज मोर सॉल्यूट विल बी प्रेजेंट इन दिस. सॉल्यूट इसमें प्रेजेंट होगा तो मूवमेंट किधर होगा? इसका सॉल्यूट पोटेंशियल, water potential कम हो जाएगा and inside the cell the water potential will be more, so the movement will be from higher to lower concentration. तो सेल की जो वाटर है वो बाहर आ जाएगी तो सेल क्या हो जाएगा? श्रिंक कर जाएगा. पहले सेल के साइटोप्लाज्म से वाटर बाहर आएगा, फिर क्या होगा? वैक्यूल से, फिर वैक्यूल से भी वाटर बाहर आएगा and it will shrink and this is called plasmolyzed cell. So सेल और सेल वॉल और सेल मेम्ब्रेन के बीच में ये जो स्पेसेस प्रेजेंट हैं, इसमें क्या प्रेजेंट होगा? The water solution will be present in here. If again we are placing this cell into the hypotonic solution, what will happen? Again it will regain its shape. The water will enter into the cell. Water will diffuses into the cell causing the cytoplasm to build up pressure against the wall and this is called turgid. Turgor pressure apply करेंगे. When अगर ये cell is placed in the hypotonic solution, again the cytoplasm will regain itself and it will create a turgor pressure. ये जो वॉल है साइटोप्लाज्म की, ये प्रेशर क्रिएट करेगी ऑन द सेल वॉल. प्रेशर विल क्रिएट ऑन द सेल वॉल. एंड दिस इज कॉल्ड टर्गर प्रेशर. And this turgor pressure is responsible for the enlargement and extension of the growth of the cell. Next is imbibition. So imbibition is a type of diffusion when water is absorbed by the solids, that is colloids, causing them to increase in volume. So you have seen in the rainy season the doors क्या हो जाते हैं? swell कर जाते हैं and it is difficult to close. Why it happens? Because of the imbibition. So what is the definition? It is a type of diffusion which takes place in solid and colloids show increase in the volume by absorbing water. So what is the example of this imbibition? The seeds, they absorb water by the imbibition. Dry wood, the pressure that is produced by the swelling of wood has been used to prehistoric man to split rocks and boulders. हिस्टोरिक टाइम में क्या करते थे, रॉक को ब्रेक कर देते थे एंड दे पुट वुड इन दिस, इसमें वुड को डाल के एंड दे एडिंग वाटर, इससे क्या हो जाता था, सो दैट दे कैन ब्रेक द वुड, दे कैन स्प्लिट द रॉक. अगर इम्बाइबिशन नहीं होता तो सीडलिंग्स के अंदर इट विल नॉट ग्रो एंड दे कैन नॉट इमर्ज आउट ऑफ द सॉइल इनटू द ओपन. दे प्रोबेबली वुड नॉट हैव बीन एबल टू स्टैब्लिश. अगर सीड में इम्बाइबिशन नहीं होता तो सीडलिंग भी it is not possible. Because of the imbibition, the seed can absorb water. This imbibition has the affinity between the solid and liquid. It moves according to the concentration gradient. So that's all for today. In the next class, I'll explain about long distance transportation of water. Thank you, and please do like, share and subscribe to my channel. If you have any doubt in this, you can ask in the comment box. Thank you.